Cell Biology. Comprehensive Guide for First-Year Medicine Students. Introduction. Welcome to this comprehensive guide on cell biology, designed specifically for first-year medicine students. Cell biology is the foundation of all medical sciences, as understanding cells is essential to understanding human physiology, pathology, and disease processes. In this educational video, we will explore the structure, function, and molecular organization of cells, from basic cell theory to complex molecular mechanisms. Chapter 1. Cell Theory. Cell theory represents one of the most fundamental principles in biology and medicine. In 1839, Matthias Schleiden and Theodor Schwann proposed that the body is composed of cells, that cells have their own life, and that the life of an organism depends on the life of its cells. In 1858, Rudolf Virchow added the crucial principle that all cells arise from pre-existing cells. George Emil Palade revolutionized cell biology using electron microscopy. He discovered ribosomes, described mitochondrial structure, and explained cell secretion mechanisms. His work earned him the Nobel Prize in 1974. Chapter 2. Prokaryotes and Eukaryotes. Cells are classified into prokaryotes and eukaryotes. Prokaryotes are unicellular organisms like bacteria that lack a true nucleus. They have a nucleoid region with circular DNA, divide by binary fission, and have no membrane-bound organelles. Their cell wall contains muramic acid, which is targeted by antibiotics like penicillin. Eukaryotes are more complex cells found in animals, plants, fungi, and protists. They can be unicellular or multicellular. They have a nucleus surrounded by a nuclear envelope containing linear chromosomes. Eukaryotes undergo mitosis and meiosis, have membrane-bound organelles, and use the cytoskeleton for movement. The human body contains approximately 1 quadrillion cells. Red blood cells and glial cells are the most numerous. The mean cell size is 20 to 30 micrometers. The law of volume constancy states that organ size is determined by cell number, not cell size. Chapter 3. Chemical Composition. Cells are composed of chemical elements classified as macroelements, microelements, and oligoelements. Macroelements include carbon, hydrogen, oxygen, and nitrogen, which form the backbone of all organic molecules. Microelements include phosphorus, sulfur, chlorine, sodium, potassium, calcium, and magnesium, essential for metabolism and signaling. Oligoelements like iron, iodine, and zinc are present in trace amounts but are crucial for health. Water comprises 70% of cell mass and is the universal solvent. Proteins comprise 15% and perform virtually all cellular functions. Nucleic acids comprise 7% and carry genetic information. Carbohydrates comprise 3% and provide energy. Lipids and ions comprise the remaining 5%. Chapter 4. Cytoskeleton. The cytoplasm contains protein filaments that form the cytoskeleton. Myosin filaments are thick motor proteins that generate force. Actin filaments are thin, 7-nanometer filaments involved in cell movement and division. Intermediate filaments provide mechanical strength and vary by cell type. Microtubules are 25-nanometer hollow tubes that serve as tracks for transport and form the mitotic spindle. Chapter 5. Cellular Motility. Cellular motility is based on different molecular systems. Muscle contraction uses actin-myosin interaction in sarcomeres. The sliding filament mechanism causes muscle shortening. Amoeboid movement allows white blood cells to migrate through tissues by extending pseudopodia. Exoplasmic transport in neurons uses microtubules as tracks. Kinesin motors move cargo in anterograde transport toward the axon terminal. Dynein motors move cargo in retrograde transport back to the cell body. Cilia and flagella have a 9+2 arrangement of microtubules. Dynein arms slide adjacent doublets, causing bending. Cilia move mucus in the respiratory tract. Flagella propel sperm cells. Chapter 6. Cell membranes. Biological membranes are composed of lipids, proteins, and carbohydrates. Phospholipids form bilayers with hydrophobic tails facing inward and hydrophilic heads facing outward. The fluid mosaic model describes membranes as fluid lipid bilayers with embedded proteins that can move laterally. The glycocalyx is a carbohydrate-rich layer on the cell surface that protects the cell and provides recognition sites. Receptors are proteins that bind signaling molecules and trigger cellular responses. Transport across membranes occurs through passive and active mechanisms. Passive transport includes simple diffusion, facilitated diffusion, and osmosis, which do not require energy. Active transport requires ATP to move substances against their concentration gradient. The sodium-potassium pump maintains ion gradients essential for nerve function. Endocytosis brings large molecules into the cell by engulfing them in vesicles. Exocytosis releases substances by fusing vesicles with the plasma membrane. Cell junctions connect cells in tissues. Desmosomes provide mechanical strength. Tight junctions seal cells together and prevent passage of molecules. Gap junctions allow direct communication between cells. Chapter 7: Nucleus. The nucleus is the control center of the cell, storing genetic information and directing cellular activities. The nuclear envelope is a double membrane with nuclear pores that regulate molecular passage. Chromatin is the complex of DNA and proteins. Euchromatin is loosely packed and transcriptionally active. Heterochromatin is tightly packed and inactive. The nucleolus synthesizes ribosomal RNA and assembles ribosomes. Chapter 8: Chromosomes. Chromosomes carry genetic information. During cell division, they condense into visible structures consisting of two sister chromatids joined at the centromere. The P-arm is short and the Q-arm is long. Telomeres protect chromosome ends. Humans have 46 chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Chromosomal abnormalities include aneuploidy, such as Down syndrome with trisomy 21, and structural abnormalities like deletions and translocations. Chromatin is composed of DNA wrapped around histone proteins forming nucleosomes. This beads-on-a-string structure is further coiled into higher-order structures, ultimately forming condensed chromosomes. Chapter 9: Cell division. The cell cycle consists of interphase and mitosis. Interphase includes G1 phase for cell growth, S phase for DNA replication, and G2 phase for preparation for mitosis. M phase is when the cell divides. Some cells enter G0 phase, a quiescent state. Checkpoints regulate the cell cycle. The G1/S checkpoint checks cell size and nutrients. The G2/M checkpoint checks DNA replication. The spindle checkpoint ensures proper chromosome attachment. Mitosis produces two identical daughter cells. Prophase condenses chromosomes and forms the spindle. Metaphase aligns chromosomes at the equator. Anaphase separates sister chromatids. Telophase reforms nuclei. Cytokinesis divides the cytoplasm. Cyclins and cyclin-dependent kinases regulate the cell cycle. Different cyclin-CDK complexes control different phases. Meiosis produces gametes with half the chromosome number. Meiosis I separates homologous chromosomes. Crossing over during prophase I creates genetic diversity. Meiosis II separates sister chromatids, resulting in 4 haploid cells. Chapter 10: Endoplasmic Reticulum. The endoplasmic reticulum is a network of membrane-enclosed spaces. Rough ER is studded with ribosomes and synthesizes proteins for secretion or membrane insertion. Proteins are folded and glycosylated in the ER lumen. Smooth ER lacks ribosomes and synthesizes lipids, detoxifies drugs in liver cells, and stores calcium in muscle cells. Transport vesicles carry proteins and lipids from the ER to the Golgi apparatus. Chapter 11: Golgi Apparatus. The Golgi apparatus consists of stacked cisternae. The cis face receives vesicles from the ER. The medial region modifies proteins. The trans face sorts and packages proteins into vesicles. The Golgi modifies proteins by adding or removing chemical groups, sorts them to their destinations, and packages them for transport. Chapter 12: Cell secretion. Constitutive secretion is continuous and releases proteins like extracellular matrix components. Regulated secretion occurs in response to signals and releases hormones, neurotransmitters, and enzymes. Chapter 13. Lysosomes. Lysosomes contain hydrolytic enzymes that break down proteins, lipids, nucleic acids, and carbohydrates. These enzymes are active at acidic pH around 4.5 to 5. Proton pumps maintain this acidic environment. Lysosomes digest material from endocytosis, break down old organelles through autophagy, and release enzymes outside the cell. Primary lysosomes are newly formed. Secondary lysosomes are actively digesting material. Residual bodies contain undigested material. Lysosomal storage diseases result from enzyme deficiencies, causing substance accumulation and cell damage. Examples include Tay-Sachs disease and Gaucher disease. Chapter 14. Peroxisomes. Peroxisomes contain oxidative enzymes that produce hydrogen peroxide. Catalase breaks down hydrogen peroxide into water and oxygen. Peroxisomes break down fatty acids through beta-oxidation, synthesize lipids like plasmalogens, and detoxify harmful substances. Zellweger syndrome is caused by defective peroxisome biogenesis, leading to accumulation of very long-chain fatty acids. Chapter 15. Mitochondria. Mitochondria are the powerhouses of the cell. They have two membranes. The inner membrane is folded into cristae, increasing surface area for ATP synthesis. The matrix contains enzymes for the citric acid cycle, mitochondrial DNA, and ribosomes. The electron transport chain in the inner membrane transfers electrons from NADH and FADH2 to oxygen. It consists of 4 complexes that pump protons into the intermembrane space, creating a gradient. The chemiosmotic theory explains that ATP synthase uses the proton gradient to synthesize ATP from ADP and phosphate. This is oxidative phosphorylation. Mitochondria replicate by dividing and contain their own DNA and ribosomes, supporting the endosymbiotic theory that they originated from ancient bacteria. Mitochondrial diseases affect tissues with high energy demands, causing muscle weakness and vision loss. Chapter 16. Extracellular matrix. The extracellular matrix provides structural support and influences cell behavior. Collagen provides tensile strength. Elastin provides elasticity. Proteoglycans attract water and resist compression. Fibronectin and laminin help cells adhere. Defects in ECM components cause diseases like Ehlers-Danlos syndrome with hyperelastic skin and Marfan syndrome with cardiovascular problems. The basal lamina underlies epithelial tissues and is composed of laminin, type 4 collagen, and proteoglycans. Chapter 17. Cellular recognition. Cellular recognition allows cells to identify and interact with others. Cell surface glycoproteins and glycolipids serve as recognition markers. Immune cells distinguish self from non-self to attack pathogens. Chapter 18. Cell aging and death. Cell aging involves accumulation of damaged proteins and organelles, telomere shortening, and decreased function. Necrosis is uncontrolled cell death causing inflammation. Apoptosis is programmed cell death that eliminates damaged cells without inflammation. The cell shrinks, the nucleus fragments, and phagocytes engulf the remains. Chapter 19. Central dogma. The central dogma states that genetic information flows from DNA to RNA to protein. DNA is a double helix with complementary base pairing. Adenine with thymine, guanine with cytosine. DNA replication copies DNA before cell division. DNA polymerase synthesizes new strands using each original strand as a template. Replication is semi-conservative. Transcription copies DNA into RNA. RNA polymerase synthesizes RNA from a DNA template. In eukaryotes, pre-mRNA is processed by removing introns and adding a cap and poly-A tail. Translation decodes mRNA to produce protein. Ribosomes read codons, and tRNA brings amino acids to form a polypeptide chain. Retroviruses like HIV use reverse transcriptase to synthesize DNA from RNA, showing information can flow from RNA to DNA. Chapter 20. Cancer. Cancer cells divide uncontrollably, invade tissues, metastasize, evade apoptosis, and stimulate angiogenesis. Oncogenes like RAS and MYC, when mutated, cause cancer. Tumor suppressor genes like p53 and Rb normally prevent cancer. When inactivated, cancer develops. Targeted therapies like imatinib and trastuzumab specifically attack cancer cells. Conclusion: understanding cell biology is essential for medical students because it provides the foundation for understanding physiology, pathology, and disease. The cell is the basic unit of life, and all diseases affect cells. By understanding how cells work, we can better understand how diseases develop and how to treat them. Thank you for watching this comprehensive guide to cell biology. Good luck with your studies.